Temperate bay ecosystems are valued for being both diverse and highly productive ecosystems. Specifically, we looked into an intertidal community. These areas often experience a lot of stresses that lead to degradation of habitat, especially because of human activities, including pollution and overfishing, as well as our focus on nutrient input.
Nutrient input is caused not only by anthropogenic factors, but also by upwelling. The area for our experiment, Bodega Bay, experiences seasonal upwelling.
Excess nutrients in the water can quickly trigger a cascade of negative events. It will start by transforming the marine plant community composition from slow-growing species that usually remain planted in the sediment to those that are fast-growing and float along the surface through the water column.
The accumulation of biomass that results by these opportunistic algae consume oxygen, creating hypoxic areas, and block sunlight from primary producers below. This leads to a loss of species diversity and loss of habitat complexity.
We decided to use a type of shellfish algae called Ulva expansa and a few of its known herbivores to see how they can impact and hopefully even regulate a harmful algal bloom. We collected Ulva from the tidal flats and separated it into 2 batches, one in ambient seawater and one in seawater with added nutrients. While the Ulva was left to enrich, we observed the herbivores in the tidal flats. Once enrichment was complete, half of the enriched Ulva was used in the field and the other in the lab trial. The same was done with the bin of non-enriched Ulva.
After enrichment, the Ulva was spun to remove excess water and divided into 5-gram portions to be distributed throughout our lab and field experimental units. We used phytoplankton nets to collect amphipods from algal mats of Ulva. We then placed the amphipods of various species in nylon mesh bags. We used Pachygrapsis crassipus collected from tidal flats. We placed 1 crab in a fiberglass window screen cage with Ulva. Lastly, we used brown turban snails, which we collected from Horseshoe Cove. We placed 3 snails in a fiberglass window screen cage with Ulva. We placed each experimental unit plus a control for each cage type on a 2-meter rope. The 5 ropes with enriched Ulva and 5 ropes with ambient Ulva were placed in the field where they remained for 4 days. In addition, we conducted the same experiment in the laboratory. Rather than cages, we used varying sizes of plastic containers placed in seawater flow-through tanks. We housed crabs in larger containers, whereas the snails and amphipods were housed in smaller plastic containers for 4 days.
As we predicted, on average, the animals preferred the nutritious, enriched algae over the regular kind. We also found that the Ulva given nutrients grew more. When we recollected the Ulva, we found that the herbivores had actually eaten some. The crabs generally eat the most, then snails, and last amphipods. Didn't expect was that our window screen cages, commonly used in similar tropical experiments, did not keep out the amphipods. Overall, one big takeaway from our experiment is that amphipods seem to be an essential consumer of algae in temperate marine tidal flats.